In this study, we will introduce you to Lifeflow, our tool for analyzing temporal categorical data. We will first teach you about the data and how to use the tool. Then we will ask you to perform some tasks with the tool. The data that we will use in this study are patient transfer data, which are based on real data from a hospital. There are several events in this data that you need to know about. We will associate the color with the type of event. Arrival in blue is when the patients arrived at the hospital. Emergency in pink is when the patients went to the emergency room. ICU in red is the intensive care unit where the patients were transferred to when they were very sick. Floor in green is a normal bed. Discharge alive in light blue is when the patients left the hospital alive, and die in black is when the patients died. The raw data look like this. This is one patient with patient ID 458-51737. The data consists of series of time and type of event. This patient arrived and went to the emergency room, was later transferred to the ICU, transferred to floor, moved to another floor, and left the hospital alive. This first visualization is called Lifelines 2. We tried to make it easier to read, so we placed color triangles on the timeline to represent events. For this example, you can see that this patient arrived, went to the emergency room, then went to the ICU, floor, transferred to another floor, and discharged. The vertical position does not mean anything. It is used to distinguish between events which occurred at the same time. This visualization has the same meaning with the compact visualization in the bottom. We just split them by type of events to be able to include the labels and reduce overlapping. Now I remove the raw data. Could you explain from the visualization what happened to this patient? This is another patient. Could you explain from the visualization what happened to this patient? What happens when there are many patients? In this example, you see 4 patients, which we displayed by 4 rows. Imagine a larger data set with thousands of records. You cannot see all of them on the screen at the same time, and it is difficult to see the overview of the database to understand the big picture. Therefore, we developed a new visualization called Lifeflow, which provides a summary of the data. We will demonstrate how to construct Lifeflow with this video. In this example, we have 4 records, 4 rows. We want to merge them into one picture that gives the overview. We started by looking at the sequence. All patients started at the blue event, so we grouped them together and merged them into a bar. After that, all of them have the pink event, so we group them together, calculate the average time from the pink event back to the blue event, and draw a bar at the average time. After the pink event, 2 of them have the red events, so we group them, calculate the average time, and draw a bar. We also group the 2 dark green events and convert them into a bar. We do the same thing for the rest of the data. Please note that the last dark green event is not grouped together with the other 2 dark green events because they do not have the same parent, meaning that the 2 dark green events on the bottom follow the pink event, while the dark green event on the top follows the red event. By now, we have shown you the transformation from raw data to Lifelines 2, and then how we construct Lifeflow. Let's have a look at an example of Lifeflow. If I ask, what is this sequence? The way to read it is to read the colors. You can see from the legend in the bottom left for the meaning of each color. So this sequence is arrival in blue, emergency in pink, and ICU in red. How about this sequence? The height of the bar tells the frequency of that sequence. For bars with the same parent, for example, the pink bar and green bar in this screen have the same parent, which is the blue bar, indicated by the yellow arrow. The most frequent sequence, or the tallest bar, is placed on the top. So you can see that patients were transferred from arrival to the emergency room more frequently than arrival to floor because it is placed above. For this one, what happened most frequently after arrival and went to emergency? Another thing that Lifeflow can tell you is the average time between events. The horizontal gap means the average time. In this example, patients can go to the emergency or floor after arrival. But if I ask which transfer is faster, arrival to emergency or arrival to floor, you can check the horizontal gap and answer that emergency is faster. How about this one? Patients arrived, went to the emergency, and then went to ICU or floor. Were they transferred from emergency to ICU faster than from emergency to floor? Please note that the most frequent pattern is the pattern which has the tallest bar at the end. In this case, the most frequent pattern is therefore arrived, went to emergency, transferred to the floor, and discharged alive. You have seen the screenshots. Now it is time to try the real tool. This is the Lifeflow tool, which has the same visualization that we had just introduced to you. At this point, there are two main features that we would like you to know how to use. One thing is the tooltip, which can tell more information about the sequence. When you move the mouse over, you can see the name of the sequence and how many instances, in other words, how many patients are included in that sequence. Also, you can see the time to previous events and time to route, which is the time from the beginning, in this case arrival. Another thing is zooming. You can use this slider to zoom in and out. Before we begin the actual study, let's make sure that you understood everything by asking several questions. Please don't hesitate and feel free to ask if you have any question, and let us know if you need additional guidance. Now we begin the study with some simple tasks. We would like you to explain what you do and think aloud. For example, to answer this question, I have to do the following steps. Let's continue and move on to some advanced features of the interface. Let's start with the concept of alignment. When patients arrive, they arrived at different times. To make it easier to compare records, we select a reference point, in this case the arrival time, and shift the time. The time now becomes a relative time, relative to the arrival time in this case, and it is easier to compare records. We adapt the similar concept to Lifeflow. For example, if you want to know what happened before or after the patient went to the floor, observing this figure, you will see that floor in green is scattered in two places. It could be scattered in many different places in a more complex data set. So, if we align by floor, we will select only those patients with at least one admission to floor, which means these two blocks on the screen, and use the floor as a reference point. To align by floor, I click on this combo box, select floor, and click on Align, and you get this visualization. The dashed line indicates the align event, which becomes the reference point of the visualization. The left side is what happened before floor. The right side is what happened after floor. Now you can see that before floor, they most often come from emergency room, the pink bar on the top. After that, they were often discharged alive. As you can remember, in the very beginning of the study, we presented the Lifelines 2 visualization. Lifeflow is a way to see the big picture, while Lifelines 2 is more useful for seeing each patient in detail. You can also use these two together in the tool. Before, you saw only the Lifeflow view. Now we will enable the Lifelines 2 view. There are two ways to use these two visualizations together. The first way is from Lifeflow to Lifelines 2. In case you are interested in a specific sequence, for example this one, you can click on the bar at the end of the sequence and see all records that contain the sequence in the Lifelines view on the right. These patients are highlighted. On the other hand, if you select any patient in the Lifelines 2 view on the right, it will highlight the same patient on the left. So you can see how many people have the same pattern using the tooltip.